Today we are going to start with the second chapter, that is sexual reproduction in flowering plants in unit 1. Now we have already seen various events which are involved in sexual reproduction in chapter 1. When we talk about angiospermic plants, the flower is the male reproductive structure of the plant. The flower will have male reproductive structure and female reproductive structure. The male reproductive structure consists of anther filament, that the whole structure will be called as stamen. When we talk about the female reproductive organ or female reproductive part of the plant, we call it as pistil or carpel. You can use both these terms interchangeably, or you can use these terms synonymously. Now here in this class we will see the detailed structure of stamen, that is the male reproductive portion of the flower. Now a typical stamen consists of two parts. One is anther and the other one is filament. As you can see in this diagram, this terminally located bilobe structure is nothing but anther, and this long slender stalk like structure is nothing but filament. Now this part can be attached with the flower, the proximal end you can call. This proximal end can be attached with the thalamus of the flower or the petal of the flower. Now this bilobe structure will be divided into compartments, and each compartment will be called as theca. So we can say that angiosperm anther is bilobed with each lobe having two theca, and that condition is known as dithecous. This condition is known as dithecous. Now in the transverse section of this bilobed anther, we can say that this anther is four-sided, that is tetragonal, and it consists of four microsporangia which are located at the corners of this anther. Microsporangia is nothing but pollen sacs, where the male reproductive part, that is the pollen grain, will be formed. Okay. Now when we talk about the structure of this Microsporangia or Sporangium, for singular we will use as Sporangium. It consists of various wall layers. Generally it consists of 4 wall layers. The order from outside to inside is, first one will be epidermis. Next will be endothecium. Then middle layers. And last, that is tapetum. Here we are talking about Microsporangium, where structure of a typical Microsporangium, where the pollen grains will be formed, that is the male reproductive gamete of flowering plant. So a typical Microsporangium, when we observe this in the transverse section under a microscope, we will observe it is having 4 wall layers and the wall layers from outside to inside are, first is epidermis, second is endothecium, third is middle layers, and last is tapetum. When we talk about epidermis, endothecium and middle layers, all these three layers will help in the protection of the formation, where the formation of the pollen grain will take place. These three layers serve the purpose of protection, and this tapetum will provide nourishment to the developing pollen grain. Now, as these cells provide nourishment to the developing pollen grain, these cells will have a dense cytoplasm, and they generally have more than one nucleus. That is, there can be many nucleus. So now, can you answer a question as how this tapetum cell after cell division becomes binucleate or multinucleate? How any cell will become binucleate or multinucleate? There is also presence of a group of compactly arranged homogeneous cell when the anther is young inside this microsporangium, and that is called nothing but sporogenous tissue or sporogenous cell. Now, these cells will help in the process of microsporogenesis, that is formation of the pollen grains. TS of anther. Here in this diagram you can see that these four are nothing but microsporangia. These are nothing but microsporangium. We have already talked about the structure of microsporangia, that it is having four wall layers. The first one was epidermis, the red cells you can see. The second was endothecum, and these layers are middle layers. And we also talked about some cells which were binucleate, and this is the tapetal layer. Now, when the anther is young, the sporogenous cell mass inside the microsporangium are compactly arranged with each other. After, when the anther grows and matures, the size of this microsporangium will become large, and the sporogenous cell will be separated from each other. There will be spaces between these cells. The size of these cells does not change. The size of the chamber, that is the microsporangia, gets enlarged. Now, each of these cells is now called as MMC, that is microspore mother cell. Now, these cells are able to give rise to microspore. Each cell is capable of formation of a microspore tetrad. We will talk about the process of microsporogenesis. Now, some of these MMC will also degrade, and these will be utilized by the structure itself, but every MMC is able to give rise to microspore, and microspore will be called further as pollen grain. That is the male gametophyte of the flowering plant. Now, we will see how the microsporogenesis processes. Microsporogenesis This is nothing but the formation of microspore. Microsporogenesis, if we have to define microsporogenesis, we will say that the process of formation of microspore from MMC by meiosis is called as microsporogenesis. So we will start the process. These are the MMC. This is the MMC. That is the microspore mother cell. It will undergo what? It will undergo the meiotic division. Meiotic division is nothing but the reductional division. That is, the ploidy of this MMC will be 2n, and after the meiotic division it will form a tetrad, that is a cluster of 4 cells, and all these cells will have the ploidy, that is n, that is haploid. Now we are calling this as a tetrad because it is a cluster of cells. As for some time these 4 cells will be attached with one another, like the various shapes in which they will be attached is, first one is like this, 1 this cell, 2 this cell, and 3rd this cell, and 4th on the back side, and this shape is called tetrahedral. The second one is like this, 1 2 3, 1 2 3 and 4. This is called what? Linear. And third one is first one, second one, third one. Third one is the isobilateral. This is, we divide the cell into equal 4 halves. This is 1 2 3 and 4. And the last one is the F shaped, which is very uncommon, or decussate shape. So for some time after the formation of the microspore, they will be grouped into a particular shape, and so this is called as tetrad. When we talk about the angiospermic plant, the most common cluster observed after microsporogenesis is this tetrahedral shape. Now these cells will be separated from each other. These 4 cells, which are haploid, which are now called as microspore, will get separated from each other, and now an outer and inner layer will be formed in this microspore, and it will be now called as what? It will be called as pollen. It will be called as what? Pollen. So we saw here that the MMC, that is the microspore mother cell, will undergo meiotic division, as we are aware that meiosis 1 and meiosis 2 will take place, and finally we'll be seeing these 4 cells, and these 4 cells will be the potential microspore. And after the formation of an inner and the outer layer that will be secreted by the tapetal cells, it will be called as pollen, or specifically the pollen grain. Pollen grain, that is the male gametophyte. Now we know that when the MMC undergoes microsporogenesis, that is the meiosis, we get 4 microspore. Now these are called as microspore. Now this microspore, after the formation, after the separation from the tetrad, now we are taking only one microscope. Every microspore is a potential pollen, or it will develop into a potential pollen, but we are taking only one microscope. So this microspore will have its own nucleus and a plasma membrane. Now the tapetal cells will secrete a thin membrane that is called intine surrounding the plasma membrane of this microspore. This is the intine. And this intine inner layer is made up of what? It is made up of cellulose and pectin. The tapetal cell will also secrete another layer, which is the hard tough layer, that is called the XII. Now this XII layer is made up of a compound that is sporopollenin, and this sporopollenin is the most resistant organic compound known to humankind. This sporopollenin cannot be degraded by any acid, any alkaline. It can withstand high temperature, and no enzyme can degrade this sporopollenin. Now this structure which is formed where the microspore is covered with a thin intine and a thick XII is now called as pollen grain. And the male gamete will be produced inside this pollen grain. So this pollen grain is also known as the male gametophyte. Now after the formation of this pollen grain, it will undergo what? It will undergo mitotic division. Now we are aware that when we are talking about division, we will see that first karyokinesis will occur and then cytokinesis will occur. So karyokinesis will occur and there will be formation of two nucleus. Now cytokinesis will occur, but this cytokinesis will not be equal, it will be unequal, and there will be a wall. This nucleus will come here and there will be a wall formation here, and this wall formation will divide the cell into two unequal cells. Now this large cell is made up of dense reserve material, and this is called as vegetative cell. Vegetative cell or tube cell. This small cell which is formed is known as generative cell. After some time, this wall is made up of a substance called callose. Now after some time, this cell will come in this area of the vegetative cell and it will float inside the cytoplasm of the vegetative cell, and this will be looking like a spindle shaped. So we can say that the generative cell looks like a spindle. So you can say that this is the generative cell and this is the vegetative cell. The vegetative cell is also called tube cell because the pollen tube formation which occurs from the pollen grain, when the pollen grain is landed on the stigma of the flower, it will be formed by the vegetative cell, and this generative cell will give rise to the formation of the male gamete. So after the formation of this whole structure of pollen grain, the pollen grain, inside the pollen grain, these all processes which we discussed have occurred, and the pollen grain is divided into two cells, that is the vegetative cell or tube cell or generative cell. Okay. In 60% of angiosperm, the pollen grain is released in this stage, that is the two-celled stage. Now what do you understand by two-celled stage? Two-cell stage means that the pollen is having a vegetative cell and the generative cell. So we can say that in 60% or more angiosperm, the pollen is shed from the microsporangia in 2 cell stage. But in 40% of angiosperm, it is shed in 3 cell stage. Now what does that mean? It means that this generative cell will undergo a mitotic division and it will form 2 gametes. Male gametes, it will form 2 male gametes before the release of pollen grain from the pollen sac. So the cell will be having what? It will have a vegetative cell and it will have 2 male gamete when it is shed from the pollen sac. Now after all these events, the pollen grain needs to be shed from the microsporangia structure, the anther. So the layers which we have talked, these layers will now become fibrous, that is dehydrate, and there will be release of these pollen outside the cell. Due to dehydration, these cells will become fibrous and they will be destroyed and there will be the release of the pollen, and this is called dehiscence of the anther, and the pollen grain will be released, and now this pollen grain will be reaching the stigma with the help of the process, that is pollination. Okay, we'll